Sir is presently a Ramalinga Swami faculty in the Institute of Microbiology Technology, CSI, IR Chandigarh. Sir completed his PhD in 2012 from National Arts Research Institute, Pune, and did his postdoctoral associate from 2012 to 2017, as well as research associate from 2018 to 2019 from prestigious Cornell University, USA. And sir is academically very bright. Sir got grabbed the medal for his merit and MSc degree and did his fellowship from Biotech Consortium India Limited, Department of Biotechnology. And sir also begged international student scholarship to attend AIDS Vaccine 2008 conference. And sir did his senior research fellowship from 2010 to 2012 from CSIR and presently has a Rama Linga Swami re-entry fellowship in 2019. And sir is funded by various societies and organizations from DBT, SCRB, CSIR, and sir is also actively participating in the research of SARS-CoV-2, and we are very thankful for that. Sir has very good citations of more than 1300 and H index of 17 and I-10 index of 22 as per the Google Scholar. Sir, we are very fortunate to have you here. And before handing over the charge to you, sir, I would like to mention the format of the session. The talk will be of 45 minutes, followed by Q&A session of 15 minutes. All the questions will be taken up after the talk, which will be about 45 minutes, as I said. And meanwhile, you can write the questions in the chat box. Sir, the stage is all yours.
Thank you, Dr. Anil Gupta, for such a kind introduction. I'm very honored today. I'm very thankful for inviting me to this forum. This seems to be very different in the recent days because I have not talked on such subjects before. But this is a really good opportunity to catch up with the students and the faculties. And I hope that my presentation will be of some help to you. Okay, so I will be sharing my slides. First of all, good evening, everyone. First of all, can you hear me properly?
Yes, Dr. Ringe.
Okay. So I'm going to talk about how to write the methodology section and the results section in the research paper. So my name is Rajesh Ringe, as you heard, Dr. Anil, and I'm based in CSIR Institute of Microbiology Technology, Chandigarh, since last 2 years now, and I'm actively researching into SARS-CoV-2 and HIV biology. So what I'm going to talk about is the methodology and results section. So as you all know, the research paper constitutes many sections. The first and foremost is the title. Second is the abstract, introduction, methodology, results, and so on and so forth. So all these sections has their own role to play. So for example, the title is the first one. We have to have a very precise title. And many journals actually have the word limit for the title. So, which means that you have to define your title, which will best define your research agenda in the research article, right? So after title, we have the abstract. Abstract is basically a very short summary, and it's a gist of the research paper. If somebody doesn't want to read the whole paper, but he or she should get the brief idea about what is going to be there in the paper. So that's an abstract. The introduction is how to introduce your research area, research subject, what problem you have addressed in the research paper, and how you approached your problem, and what was the premise behind that. So all that will come into the introduction. So introduction has to be a little longer, and it has to systematically introduce the audience to the subject area that you are going to write about in the next upcoming sections. Methodology is basically a section that describes all the methods that you used into the in your research proposal or in research article. For example, you have done many experiments or some surveys. What was the method basically behind those, and what tools you have used what are the techniques used, what are the equipments used, and what is basically the methods you use. And after that, we have a results section into the research paper, which will describe what are your basic findings. So the first result is this: I made this number of flows. So describe all those things. So results basically just describe the results. So this is the experiment and this is what I found. So that is the results section. There you do not argue why I found that or why I did not find this. So that is not the place to have those arguments. So there is a different section, separate section for that. That's called as discussion. So discussion section is the one where you have ample of room to defend your results. For example, you found that many COVID-19 patients actually die because they have a very cytokine storm in them, right? So this is where this is the result, and you have to discuss why it would have happened, and what is the premise of this, and whether there is other evidences which support that. So that is the discussion. And then after that, the conclusion comes. The conclusion is a short summary based on all the things you have shown above. And then there is a reference list which includes all the references that support your methodology, that support your results, and that supports your findings. And there are some certain journals which also have the appendices or the supplementary information. If you want to publish any supplementary information that doesn't go into your main text, but you think it's very important for the reader, and many readers will get the benefit. So that is supplementary information you can provide. So this is a basic architecture, or this is a basic frame of a research paper. And I'm not going to focus on each and every section, but I'm told today to focus on the methodology and the results section. So I will be focusing on these two sections. So first one is how how to write the research methodology. So you started with title, you wrote your abstract, you wrote your introduction, and then you have to write your methodology. So describe the tools that were used. For example, if you want, you have so many methods to describe actually, because you employed so many different techniques to meet your research objectives, and you have to describe them one by one. So first thing to describe is the tools. So what are the tools you use? For example, if you used any clones, or if you used any organisms such as human subjects or bacteria or virus, whatever you used, that is your tool, and that you have to describe first. So for example, I used E. coli for the expression system. So I have to describe E. coli there. What was the strain? Where did you get it? And how did you grow? What are the different characteristics? So that's the start of your methodology section. And after that, describe your methods of data collection. So how did you collect your data? Whether you did any kind of survey, or whether you used your own experiments to get the data and report the data, or you used somebody else's data and you are analyzing or reanalyzing that data. So you have to describe those methods of data collection. So how did you collect your data? Did you do your own experiments? If yes, then what experiments you did? If you used the data that was already there in the literature, so how did you collect this? So this is the method of data collection. Next is describe your methods of analysis. So how did you analyze your data? I mean, what techniques you have used? For example, you got some data like you did a survey. For example, in New Delhi, you went and you did a survey of 10 hospitals, and you found that different hospitals has different number of patients, and what is their viral load, and what was their disease manifestations. So this is basically method of analysis, and what analysis you have used and what techniques you have used. If you have used some techniques, so what are the equipments used and why those equipments? You have to justify. For example, if you wanted to do the sero survey, for example, you want to test, you want to see New Delhi has how many patients and what is the serological status of those COVID-19 patients. So you took their blood, you took their sera, and you did some experiments. So which equipments you used? You used the RT-PCR machine, or you used the ELISA reader, or you used the plate reader, and why you used those? I mean, what was the agenda there? So you have to describe that, and then evaluate and justify your methodological choices. I mean, if I want to see the antibody titers in the COVID-19 patients, I will not do RT-PCR, but I will do simply ELISA on the serum. So why you have to use that particular technique, that you have to justify, right? And then use past test. Whenever you describe your methodology, methodology is the one, it means that you have done your research and you are writing your paper. Now you wrote all the sections. So everything is done. Research is done. Now you are writing. So that is why it is a past tense. So I used this. I used E. coli to express my protein, or I used mammalian expression vector to clone my gene, something like that. So every sentence in the methodology has to be in the past tense, because the research has already been done, and now you are recalling what happened last month or last year. Now you are describing those results. So the next is tools used in the experiments and the analysis. So let's start with what is the tool basically. What are the tools? So tools are the ones that can help you in your research. For example, if you are using some organism, for example, E. coli, or for example, now COVID-19 is going on, and many people are doing research in COVID-19 or viruses. So virus is your main tool, because you want to do so many experiments. So you need to have a virus. So virus becomes your important tool. It's not the data; it's a tool, right? To study your research objectives or research agenda. So that's a tool. So if you used any clones, or you made any DNA constructs for your protein expression or antibody expression, so you used those things, and then you have to describe all those. So this is your basically the tool. If you use any organisms, for example, bacteria or virus or yeast or mouse or any human subject, so these are the organisms in your research, and they are the tools. They are not the data, they are the tools. To describe them, describe the characteristics of the constructs or organisms used. For example, you isolated influenza virus from a flu patient. So you have to describe the characteristics, why you chose that virus, and what was your objectives. So you have to justify and describe the characteristics of this organism. How were clones created? So that is also related to the tools. So for example, you use some constructs to express the antibody or to express some protein. So how those clones were created? Whether you created by yourself by using some primer pairs, or you obtained from some other investigators, that has to be described properly. What are the expression systems? Whether you use mammalian cells or yeast cells or bacterial cells or some other cells, you have to describe the expression system. So these are all the tools in your research, and you have to describe your tools properly so that the reader understand they have used this system, or they have used bacterial system or yeast system to express this, and they will know what are the drawbacks or what are the merits, and they will also look for the justification why we use them. If you use recombinant proteins, so how did you use the recombinant protein? So whether you made them into the lab, or you just bought them from a commercial agent or company, you have to describe those. So these are basically the tools used in the experimental analysis. So basically, I am from a biological laboratory science background. So my presentation is going to be heavily loaded with the experimental biological experiments. I am not a medical person. I have not done a clinical research. So so please bear with me. I was told that it has to be a little bit generalized presentation, but this is going to be a little bit skewed towards biological experimental science. So please try to understand some of the jargons or methodologies used in the presentation. So these are the tools, and you have to describe all those tools in your methodology and the analysis. So next is after tools, next is the equipments used for the experiments and the analysis. So many people think why the equipment is important. I mean, PCR machine is a PCR machine, whether it is in IIT Delhi or whether it is in some kind of university or whether it is in PGMR. So PCR machine is a PCR machine, right? But that's not the case. I mean, if you are using a PCR machine which was made 10 years ago in PGMR Chandigarh, and IIT Delhi is using a latest PCR machine, so they have a different actually characteristics. They have different detection systems. They have different sensitivities, and that is where the results can be little bit off the scale. So sometimes we see the patient comes to us and then he said, oh, I did my PCR test, COVID-19 PCR test in this hospital. I came negative, and now you are giving me a positive report. So why the equipments are important? Because the hospital might have used something else, research institution has used something else, so data cannot be properly put together. So it is important, if somebody wants to reproduce your results, it is very important to describe your equipments also, because they are important, right? Experimental conditions. So what are the experimental conditions? So for example, you did a RT-PCR test to diagnose a COVID-19 patient, and what are the experimental conditions you used, and what is the machine you use, whether you use BioRAM or whether you use Thermo Fisher. So all these things has to be documented. For example, if somebody wants to reproduce these results in their own setting, in their own lab, in their own country, then it has to be reproducible to some extent. If not exactly identical, it has to be, the results has to be similar. And also the measuring conditions. So what were the measuring conditions? How did you measure your data? Whether you use absorbance, whether you use luminescence, whether you use any other readout, that has to be properly mentioned. And it is also important to mention which version and which company of the measuring device was. For example, if you use a sequencer machine or PCR machine, whether it was from BioRAD or whether it was from Thermo Fisher or whether it was from High Media, whatever it is important, because every machine has a different sensitivities and different ramping temperatures, different ramping speeds. So the PCR results might differ, and your viral load or whatever end results you want to look for, that might change. So I use this tool, I use this equipment, and I use this measuring condition, and this, by using all these things, I found this. So if somebody wants to reproduce, he can do a little bit tweaking here and there, and accordingly they can set up their own experiments. So this was about the equipments and why they were important. And next important section is the methods of data collection. So what is your method of data collection? What are the methods of the experimental evaluation? For example, if you want to just see the antibody titers in the patients, COVID-19 patients, what will you do? In that case, simply I would take this blood samples and the serum samples, and I would do the ELISA assays, right? So this is my method of experimental evaluation. If I want to check the antibodies in the COVID-19 patients, I will not do RT-PCR, but I will do ELISA by using the antibodies on those. So this is why it's important to mention the experimental evaluation method, right? And what are the experiments you have done for evaluation of anything? For example, you used protein expression, or you did a protein characterization. So what are the experiments you have done? For example, if I want to express a protein, I made these constructs, and I transfected this construct into mammalian cells, and that is how I got my antibodies. So this is the experiment, and that's why you have to describe the method of data collection. So if I want to say I did a protein expression, what was the method of your data collection? I simply say that my method of data collection was the absorbance. I checked the absorbance of the purified protein or purified solution, and I found this microgram or milligram protein was there based on the absorbance. So this is the method of your data collection, and that's important. And what are the expression systems? If in your research paper or in your research, you have used some expression systems, for example, mammalian cells or yeast cells or bacterial cells, you have to describe, because characters can be different. Mammalian cells, the protein expressed in mammalian cells can be different than the protein expressed in the bacterial cell because of the fundamental differences on the systems. Bacterial cell is a prokaryotic system and mammalian cells is a eukaryotic system, much more advanced system. So the post-translational changes are happening in mammalian cells, but not in the bacterial cells. So the final product, the protein that you get, can be different. So that is why you have to explicitly describe what is the expression systems there. The recombinant proteins, if you have used, what is the method of getting the recombinant protein, whether it was in-house prepared or it was commercial, because the characteristics can be different. If somebody wants to reproduce your data, and if you have used the commercial source, then they can buy the same protein by using from the same commercial source. But if you have used your in-house methods, then you might have a little bit of leeway that I use my in-house protein, and that is why it may be a little bit different than theirs. And what are the readout for the experiment, whether you use absorbance or fluorescence or the imaging system, right? So if I want to do the histopathology of COVID-19 patients, and if you get a tissue of a small airway, then I would not use absorbance or fluorescence. I would just use the imaging system. I would image the tissues and see whether there are some histopathological characters there or manifestations there. So this is what is called as the method of data collection, and what are the methods used, and what is your what are the tools you have used, and how did you collect the data. So that has to be properly documented. Here on the right side, you can see that this is a pictorial form, and I want to see what is the antibody titer in the COVID-19 patients. So what I would do is, I would do the survey of, for example, 10 patients or 20 patients, and I would get the blood samples from them, and I would isolate the sera, which contains antibodies, and I would do the ELISA. So the readout here would be the absorbance. So what is the absorbance I get, and that will tell me whether the antibodies are what is the titer of the antibodies, what is probably the affinity of the antibodies, and what are their other characteristics, for example, neutralization properties. So this is the method of your data collection. So ELISA assay and the neutralization assay, for example, is my way of data collection, and the methodology to get the to see the objectives, right? So this was about the methodology, and now I'm shifting to the results section. How to write the results section, because after methodology you have to write the results. So methodology, you describe all the things. What was your methods, what was your techniques, what were your tools, and what is your method of analysis. And after describing all those things, now you are writing the results section. So how to write the results section? So you may have many results actually, 1, 2, 3, 4, 5, 6. For example, you have 5 important results in your research. Now you have to describe them one by one. So it has to be generally first come first basis, and it has to maintain the flow. For example, first you have to briefly mention the type of analysis you use. You have already described all these things into the methodology, but here again you have to remind the audience that, and very briefly, that I use this type of analysis, and I obtain this kind of results. So now first you describe this analysis, and then you start with the results section. And when you generally start with the results, then you have to write what are the experiments done briefly, and basically focus on what are your findings there. For example, if I wanted to check the serological conditions into COVID-19 patients, I would just say I collected 100 people sera, I did ELISA analysis, and then I found that this many patients has a very high titer of antibodies. For example, 70% people and 30% people did not have a very good antibody titer. So this is your finding basically. And if there are any groups, for example, you grouped your observations like mild symptoms or severe symptoms or very severe symptoms, then whether those groups has any particular characteristics. For example, I have here mild and non-mild two groups basically. Just take, for simplicity, we'll just go by the two groups. For example, mild COVID-19 symptoms and non-mild severe COVID-19 symptoms. And if there are significant differences in your findings between these two groups, then you have to say that and you have to focus on that, because that becomes the important result. And also you have to apply the statistical analysis there. You use any kind of regression, linear regression or non-linear regression. If there is any correlation you have used, for example, age versus the antibody titers, and you find that younger people have lower antibody titers than the older people. So that is your correlation basically, right? So if there is any kind of correlation, what is the significance of that correlation? How strongly your data correlate with your claims? So that has to be properly and statistically backed up. Then you have to give the p values. What is the probability value? What is the r value? What is the strength of your correlation? So all these statistical parameters you have to give in your results. You have to mention the p values. You have to mention other statistical parameters, and also you have to give the concise summary of each result, including relevant descriptive statistics, statistical means and standard deviations. So it's a large number of data, for example, you have, and whether there is and deviations are always there, because this is laboratory methods, right? There are always deviations, and that is why the deviations are important, and you have to mention what is the standard deviation in your data collection, and what is the inferential statistics, for example, p values, r value. These numbers are often placed in parentheses. If you make a figure there and you have a diagram, and then on the diagram you used you just mention in parentheses, in brackets, what is the p value, which shows what is the strength of, what is the probability that you will find this kind of results. That's a probability parameter, and you have to mention that. That gives a strength to your data. And also you have to give a brief statement of how the results relates to the questions, right? So what was your research agenda? I mean, my research agenda, for example, was whether and how antibody titer will differ between the patients. So that is why you collected different patients and you obtained different data, and you found these things. For example, in the mildly infected people, you see less titer of antibodies versus to those which were older people, right? So this is the result, and this is the statement you have given in your results. And how does that relate to your research agenda? Yes, my research agenda was the same. I wanted to check what is the serological status of the COVID-19 patients. So that is why you have to briefly remind in the results, how does it correlate with your research agenda, or research question in general. So that's the general way of writing the results. And here, for example, I have given a data. You analyze, you got these results. For example, it's very general results I presented here. There are different vaccines available. On the x-axis, you see Moderna, BioNTech, Novavax, AstraZeneca, Johnson & Johnson, Inovio, and then there is an in-house that we were working on. So the blue bar is showing that what is the, when you tested your vaccine in mice, what was the neutralization titer you got on the y-axis. You can see on the y-axis in the log scale, you have mentioned the neutralization titer, means the antibody titer you got. So all the blue bars are for the mice, and all the red bars are for the COVID-19 people. HCS means human convalescent sera, and the green one is the ratio of the mouse titer versus the human titer. So this is the comparison. If you want to show the comparison of something with something, for example, I'm showing, for example, I'm showing the comparison of Moderna vaccine versus the AstraZeneca vaccine. So I want to show it in a diagrammatic way. So I just made the bar graph here, because it's a very easy way to show what is the antibody titer between or among the vaccines. So the best one here you see in the mouse is the Novavax, which is the longest bar, the blue bar. So then the results, you can say the Novavax vaccine makes the antibody titer, which is so far the highest among all the vaccines. So that is your main claim or main result or main finding of the results. And how does that compare with your own vaccine? For example, in the last one, which I highlighted into the dotted bar line, dotted box, is our in-house vaccine. So this is just for example. I'm saying that my vaccine, which we made the vaccine, it is showing as good the antibody titer as Novavax company had. So our vaccine, for example, is showing a very good titer, and it's comparable to the Novavax, and it's comparable to other vaccines, right? So this is your result, and this is the way of your presentation of the data. So I thought for this kind of data, the bar graph is the best way. There I can show the comparison between all the vaccines so far, and it's very easy to see for the reader, right? So this is the way of writing and describing the results. You can also describe your results by, and this is very essential part of the research actually. If there are so much of data, you can't have the figure for every data, right? You can't have the figure. You may end up, if the research is very huge, and there are many investigators are involved, there are many patients are involved, and many parameters are involved. You cannot have the figure for each and every single thing. You can each and every single experiment, right? You don't have to necessarily say that well, I did my experiment on the last Sunday, and then I came back to home, something like that. You don't have to describe all those details. So if you have so many figures, like 100 figures, you can't show all those hundreds, because then that will exceed the limit of the research paper, and journal will not want to publish your data because it's too big. You have to make it very concise, and you have to make it very readable, right? So what are the different tools we have then? We can use the graph, or we can use the table. So tabulating data is a very easy way to present the data. If there are so many parameters, for example, COVID-19 patient data we analyze, and you have like what is the age of the patient, what is the gender of the patient, what was the CT value of the patient, what was the viral load, what was the antibody titer, what was the neutralizing antibody, so many other parameters you have checked. So you can't have the figure. Best way is to tabulate that data. So for example, here I'm showing salt concentration and light transmittance. It's a very simple experiment you have done, and you have taken the solution of a solution having different salt concentrations, 0, 3, 6, 9, and so on and so forth. And you did this experiment 5 times, for example. So you can't have a figure for everything, but you can actually tabulate that data, right? So here I have tabulated that data for you, and then there are different trials, trial 1, 2, 3, 4, and I see the transmission and at the different salt concentrations. The other way to represent this data is also by using a line graph. For example, on the right side, I have shown transmittance on the y-axis and the concentration of the salt on the x-axis, right? At different concentration of the salt, what was the transmittance? So you get a line graph, and you can see the trajectory, how the transmission actually is increasing with respect to the concentration of the salt. So that is also one way to show. So you have to choose what is the best way of presentation. For example, if you think that this data can be presented nicely in a figure, and that will be much more readable, that will be much more understandable, then just present a line graph. Or if you think that this is too much of data and then reader will not be able to get his eyeballs on each and every figure, so best way is to just tabulate it. So if the reader is interested in each and every figure, he will go through the table and he will analyze himself what is going on here, right? So this is the best way to present your results, whether either it is in a table, in a tabular form, or it is in a graph. So it depends what you want to show and what is the importance of that data. And that's why I'm saying that you have to choose a relevant graph to present the data. What is the relevant graph? Whether it's a, if you think it's a line graph is better suited, or the bar graph is better suited, or the column statistics is better suited. So you have to choose all those things based on your need and based on your agenda. Again, for example, I'm here showing this is I just put here my own data here just to explain you how I will be doing my experiments and how I will be actually presenting my data. So on the left side, you see there is a line graph, and then these numbers, different colors of the lines. You see different sera from the different COVID-19 patients. Now I wanted to see what is the neutralization properties, or what is the neutralization, whether the antibodies in the patient, COVID-19 patient, can neutralize the SARS-CoV-2 virus. And that is why I got a lot number of sera on the patients. For example, here I'm showing 20 sera. So I got sera from 20 patients, and I have done the neutralizations, and I have shown the percent neutralization on the y-axis, and on the x-axis I have shown that what is the dilution. So here in this experiment, I wanted to see what is the neutralization title, how best your antibodies can neutralize the virus. That's what I wanted to see here. So this is the data only for the 20 sera, 20 patients. And for example, and this is just a one variant, Wuhan variant. And if you have like 10 different variants, and if you have 1,000 sera to assess or evaluate, you cannot have it. It will be impossible for you to have the graphs for all those. And the reader, for example, you wrote a paper. If I write a paper based on this, and I present it to somebody else, he or she may not be able to see all those graphs, or or the reader will quickly lose the interest. Oh my God, there are hundreds of graphs. I can't see each and everything. So here, what I would do is I would just tabulate my data in a nice long table, and I would have a different viruses and the different sera, and I would just make a table. So for example, the table shows what is the serum number, 22, 23, 24, all the sera number, and what is the ID50 values for each serum. So that's the best way actually. And if the reader is in analyze, then he would also understand what are the different titers in the different patients, right? So it is better to tabulate when the large number of graphs are available. So if your research agenda was big and there's a large number of data, so nowadays we call it as big data, because clinical data are sometimes the big data, because clinicians tend to study large cohort of the patients. For example, 2,000 patients, 3,000 patients. So they have to choose a best method to present that big data in a small, concise manner, so that reader also gets the basic message from the take-home message from the figure or from the table, and also digest all the data quickly and in an understandable manner. So you have to find out what is the best way to present your data. So this is my experiment, and I would here not choose presenting the graphs on the left, but here better would be for me to present this data into the tabular form. So I would go with a table here. So, and then what are the tables and the figures? In experimental research, it is helpful to include visual elements such as graphs and tables, right? So you are writing your results basically, but you can't write each results for 10 pages, right? You have a definite limit of words, and you have to describe your findings within that limit. Therefore, it is very useful to have a representation, visual elements such as graphs or the tables or some schematic diagrams. Like if you want to show the immunization schedule, you don't have to describe you injected on this day, and then you followed on next month, and again booster gave on the next month, and then medicine started on July, and then again the booster medicine started on 3 months later. So all those things cannot be described in the words properly, because the reader will not be able to understand, will not be able to get and digest. But here, the diagrammatic representation, a schematic diagram would be very important. You can just show in a line schematic representation, or in some kind of nice cartoon shape, cartoon style figures, that this is the scheme of your immunization, this is the scheme of your medicinal regimen, and this is what you have done. So that will be the part of the figure, and and it will be good for the reader to just read that, see that, and then get the idea what has happened actually, what was their immunization regimen. Not all small observations needed to be prepared as graphs. You have to present only the big and very important figures and important findings. You can't describe each and everything. For example, you don't have to necessarily describe that I have done these experiments in the last month, and I was not in my good shape that in that month. I was so busy, I was very stressed. You don't have to describe all those things, because that's not relevant for the audience. But you have to describe a very important findings. You don't have to describe each and every small details. Refer to all tables and figures in the text, but don't repeat the information. So there should not be any kind of redundancy. If you have described something into the introduction, don't repeat the sentences. You have to be very specific about the limit, very particular about the limits and space given to you. So refer to all tables and figures in the text, and don't repeat the information. Make sure that you refer. If you have given like 10 figures, you have to have a reference for each and every figure. Make sure that every figure has got you have given justice to each and every figure, and you have described them in particular in enough details, so that reader can trace. Okay, now I want to see the figure number 9, because figure number 9 is very important to me, because it has shown the effect of the vaccine on the COVID-19 death rate. That's the important figure for a reader, for example. So he would just go to that figure 9 and then look for the description in the paper where it is described. So something like that. So make sure that you have described each and every figure in a precise manner. Not all the details given, because it is not needed. Because the small details can differ from lab to lab, country to country, and setting to setting. For example, here, for example, you will use a different reagent, and some other laboratory will use a different reagent, but as long as it's the same chemical, it's going to be fine. You don't have to describe all the small details. Then the text should summarize or elaborate on specific aspects or tables, and not just restate or same numbers, restate the same numbers that you have already presented. So if there are figures and there is a description to the figures, there should be some kind of coherence, right? You need to properly elaborate specific aspects of your figure, and not just say that what reader can see on the figure. You don't have to necessarily say the same thing again, but you can choose the different words, different language, and make your point, right? it shouldn't be looked at like it's a repetition of the writing, or it's a repetition of statements. Give your tables and figures clear descriptive titles and label so the readers understand what is being shown. You have to label your figures properly, very properly. This is the label of the figure. This is the title of my table. So the reader can understand, even if he doesn't have a time to go through the text. He doesn't have time, for example, to read all the paper at that particular moment, but he thinks, oh, it's very important to just glance over the data. So he would look for the data, the figure, and you need to have a legend under the table or under the figure. So he would just, or she would just read the legends. So there should be proper title to that figure. There should be proper description to that figure, and that should be enough to get the basic idea what is happening on with that result, right? And that is all about the research and the research methodology and the results section. So methodology is the basic description of all the methods, descriptions, analytical methods, and the equipments and basic tools in the methods and in the research. And the results section describe all the results that you have opted. And after that, your discussion will come, where you will have the opportunity to discuss your results. And you can also counter, or you can also support your own findings and counter others' findings, so something like that. So that's a different section of these results. And I hope that you have understood what I have said here so far. So, and that's all from me. Thank you for your attention. Thank you very much for your time.